Good afternoon and welcome, my microbiology students. We are going to be covering Chapter 3 today. So in Chapter 1, we discussed some very basic microbiology concepts like what are microbes. We talked about a little bit of taxonomy and how we classify microbes. And so now we're going to go into further depth into bacteria. So Chapter 3 is titled Bacteria and Archaea, the two of the three domains of life. And the way that I'm going to teach this chapter is I'm only going to go over sections 1 through 4. So 3.1, 2, 3, and 4. I don't feel the need to cover sections 5 and 6 because I teach this course in a clinical approach. There is no clinically significant organisms from domain Archaea, and I don't feel the need to cover section 6 either. So we'll cover bacteria in overview. We'll talk about their external structures. We'll talk about the cell wall and membranes, and we'll also talk about the internal structures that are found within bacteria. So to start, bacteria and technically archaea are prokaryotes, and they're different from eukaryotes. These are our two main cell types, prokaryotes and eukaryotes. And the difference between the two, when we get down to the just very basics, is eukaryotes have what we call in a cell a nucleus. And our prokaryotes do not. As a matter of fact, if you break the word down, pro means before and karyo means nucleus, before nucleus. If we take a look at the evolutionary tree of life, we have I'm going to write E for eukaryotes, we have our A, the archaeans, and then we have our B, bacteria. Both of these two domains include organisms that are prokaryotic, which means they do not have a nucleus. Nuclei are only found in organisms that are in domain Eukarya. So, essentially, the difference then is how their DNA is packaged. The eukaryotic organisms that have a nucleus has DNA housed there. So in eukaryotic organisms, DNA is packaged into the nucleus. And it's also wrapped around histone proteins, but that's a little more detailed than what I really wanted to go into. Number 2, another difference between prokaryotes and eukaryotes is the makeup of their cell wall. So the next word here that's underlined is peptidoglycan. This is a polymer that is found in bacterial cell walls. Other types of cells can have cell walls, even in the domain Eukarya, but not all of them. So for example, animal cells. Animals are in domain Eukarya. Our cells don't have cell walls. Some other organisms, though, like fungi, they are eukaryotic organisms and they do have cell walls. But bacteria specifically has peptidoglycan in their cell walls, and that's extremely important, and you'll see why soon. We're going to talk about gram staining at some point in the class, and it's reliant on that fact that it has that peptidoglycan-filled cell wall. There are other unique chemicals that bacterial cell walls consist of, and we'll get to more details on that later. But for now, peptidoglycan is very important. Additionally, bacteria have internal structures that are different compared to our eukaryotic cells. So in eukaryotic cells, they have lots of what we call membrane-bound organelles. And that's just essentially a fancy way of saying that these cells that are eukaryotic have these little tiny, what we'll call them tiny organs. It's like the cell itself has organs, just like your entire body has organs that do a specific function. Like your heart pumps blood, your brain allows you to think. So in this case, organelles in cells are structures that do specific jobs for the cell. And our prokaryotic cells, including bacteria, don't have very many organelles. They lack these membrane-bound organelles. So, again, cells are the basic unit of structure and function of all living things. We have two types of cells, prokaryotic and eukaryotic, and the difference essentially is whether or not they have a nucleus. So here in the eukaryotic cell, you can see right there dead center that nucleus, which is considered essentially the command center. It's almost like the brain of the cell, if you will, is right there in the center. Notice on our bacterial cell, our prokaryotic cell here, there is no nucleus. That DNA is just kind of free flowing in this area, which they call the nucleoid. Don't confuse that with nucleus. Nucleus is membrane bound. You can think of this as like there's a membrane there. It's holding in the DNA in that spot. All right, so you can see all the spaghetti looking stuff here. That's DNA. And the membrane bound nucleus is encapsulating it. We don't see that with bacterial cells. Also, you're going to see lots of other structures here. We won't go into them right now because we're going to talk about them in just a moment. But these are all organelles, like mitochondria, vacuole, Golgi apparatus, peroxisome, smooth endoplasmic reticulum, and so on. And again, notice our bacteria cells, they don't really have that. They don't have those organelles. There's very few of them. These also usually have a cell wall that contains, once again, big important word, peptidoglycan. Alright, so let's look at the structure of bacterial cells. So this picture comes from your book, and it shows what is in the outside of the bacteria. So notice the pink highlight is the external part of the bacteria. What's in the boundary of the bacteria? And then what's the stuff on the inside? So you'll see a list of items. Externally, some bacteria cells can have flagella, pili, fimbriae, nanotubules, nanowires. There can be surface layers, S layers, glycocalyx, long lists here. But do know that not all bacteria cells have all of these. So some bacteria cells have an outer membrane. All of them do have a cell wall, or I'm going to backtrack that. Not all of them do, but most of them do. A cytoplasmic membrane, all of them have that. All of them have cytoplasm, ribosomes, and the list goes on. I won't spend too much more time on the slide because we're going to talk about these organelles individually. And that brings us to another picture from the book, which is huge, and it has lots of examples of of structures found in bacterial cells that we're going to go over. I know this writing, you can't see it. I just want to demonstrate the picture. It's from your book, and we're going to go over each one individually so you can see it at another time. So, bacterial shapes. Bacteria can come in a variety of shapes. So many bacteria function as independent single-celled unicellular organisms. Uni meaning one and cell meaning cell. And they have an average size of approximately 1 micrometer. So if we look at the shapes that are listed here to the right, I know they're again difficult to see because of small writing, and again I'm going to show you them individually after this slide. But all of them are going to have various shapes, and they are going to range in sizes of usually around 1 micrometer. That's really small. Obviously, we have to have a microscope in order to view them. There is another term before we move on I want to mention, and that's pleomorphism. Some bacteria we call pleomorphic because they can have variations in their cell wall structure, and that can cause them to have slightly different shapes from one to the next. Alright, continuing in bacterial shape, the first example of a basic bacterial shape is the coccus. The coccus, or cocci, plural, is a spherical bacterium. They are shaped like a ball. Great example would be any kind of Staphylococcus bacteria. I bet we're probably pretty familiar with that, like Staph aureus, for example. But what you're seeing here are various examples of bacteria that would be considered cocci. This is a light microscope image of Staph aureus under the microscope. It almost looks like little grape clusters, all these little individual purple balls. And then we have this colorized image showing you that same image using a scanning electron microscope so we can see it even closer up. The next bacterial shape is the Bacillus, or bacilli, plural. So notice you change the US basically to an I and it makes it plural. There's also a genus of bacterium called Bacillus, so make sure we're kind of aware of that so we're not confusing what we're saying. And they are rod-shaped. Bacillus bacteria are rod-shaped. And they can be pretty varied in their actual form, but they are generally in a rod-ish design. So depending on the species, they can have blocky rod shapes, the ends can be tapered, they can have a round end, they can be filamentous, very, very skinny, like strings essentially, or they can even be club shaped. Some have drumstick shapes and so on. If we have a rod that is really, really plump, a really fat one, then we would call it a coccobacillus because it almost appears like the rod-shaped bacteria is round due to how thick it is. So comparatively, here's a rod-shaped bacterium. And here, I didn't do a phenomenal job drawing that. I'm sure many of you may not know me personally, but I'm a terrible artist. But this would be maybe considered our coccobacillus. Maybe if I went a little, I'm making it worse. Okay, we'll move on. So anyway, here's a few variations that were mentioned in rod shape. So you can see the really blocky ones here. We see some here that look more rounded. These also look a little more rounded as well. But that definitely shows you that blockiness there of the blocky rod-shaped bacteria. Another example of a basic bacterial shape are Vibrio. Vibrio essentially means it's comma shaped. So notice if you draw a comma, it looks like that. These bacteria are bent. Here we see a colorized scanning electron image of it. And here we see the same thing, but in a light microscope. So as you can see, many of these bacteria here shown have a curvature to them. And this one specifically is a Vibrio vulnificus. Next, bacteria can be in the shape of spirillums. So it might help you to remember this by saying spirillum spiral, possibly, because they do kind of look like a spiral-shaped body, like a curled piece of hair or something like that. They have a rigid, very rigid helical shape, and it's twisted twice or more along an axis. It's kind of like a corkscrew would be a great example, like a wine opener corkscrew. So these photos here are of a bacteria called Campylobacter jejuni, and as you can see, they have this corkscrew shape. And this image shows you what it would look like under a microscope in a laboratory that would use a light microscope. Next, we have spirochetes. All right, again, spiral shaped. However, their bodies are more flexible. All right, so that's the main difference here between spirochetes and spirillum is the flexibility of their of their membranes. Okay, so moving to our next shape, we have filamentous bacteria, where they kind of look like, I would say, thread-like. This shows you an illustration of one. You can see how it has these branching points, and this would be an actual image seen under the light microscope. This is a picture of a bacterium called Streptomyces. Now, next we need to talk about bacterial arrangements. So the previous shapes that we've discussed can also be classified by arrangements, how they organize themselves together. So we'll start off with our cocci arrangements. Some bacteria live singly, so they're just single cocci. Some bacteria tend to form pairs. We call those diplococci. Di means two, and then you have two cocci. They form a pair. So as you can see here, this is a single bacterial cocci. Some species reside in twos like this, Diplococci. A great example of that would be, let's see, Neisseria gonorrhea. I bet y'all can guess what that causes. Gonorrhea. All right. Some cocci reside in tetrads. That's groups of 4. Tetra meaning 4. So those tetrads can also arrange themselves in packets like this, or even linearly. Additionally, we can have irregular clusters of bacteria. So this would be an example of an irregular cluster. It looks basically like a thing of grapes. Great example of that, Staphylococcus, or even Micrococci. Streptococci are chains of cocci. So know that when you see strep means chains of something. And lastly, Sarcina. These are cubical packets of 8, 16, or more. So that's what we see here. It resides 3-dimensionally in a cube rather than a tetrad of 4 on a single plane. Most commonly, the ones you'll be seeing are probably going to be the single, the Diplococci, the streps, as well as Streptococci, or if it was a bacillus, we call it a Streptobacilli. But those other ones are also possible too. Speaking of bacillus, bacteria, depending on species, can reside as individual bacilli. They can form chains. If we have 2 rod shapes that are in a chain together, a great example of bacteria that tend to live that way are Moraxella. Here we have Moraxella bovis is what it's the genus and species. We also have cocci that can, bacilli that tend to form chains. An example of that would be the Streptobacilli that we see here. And then finally, our palisades. Palisades are cells that are in a chain, but they're partially attached by a hinge region at the ends. So instead of being able to stretch out like these Streptobacilli, they kind of look like a messed up keyboard is kind of how I describe it. But you can see they're still in a chain, but they're all bent around one another. So the example here that is given is Corynebacterium. All right. So our next section discusses external structures. So we're going to talk about the things that can exist on bacteria on the outside of them. And namely, these are called appendages, and there's different types of appendages. We'll start with the first one that's on the screen, which is flagella. So flagella are essentially these long whip-like tails. So you see this individual cell has 1 flagella. Some bacteria can have multiple flagella, and there's various different arrangements they can have. As you can see, this bacterium has multiple flagella that are all over its body. This one looks like it has 1, 2, 3, 4 flagella that are on the end of it. They're not all over its body, just on the end. And this one looks like, I believe, it has flagella on both ends. It's a little more difficult to tell in this picture. But if you look through the text, they go into a lot more detail about flagella and their arrangements and how they make microbes move. We're not going to go into all those details in our lecture, so you will not be tested on that. Remember, your exam questions come from my PowerPoints and these lectures. So we don't need to go that deep to cover our state standards. All right. Something that might be of use to know is that motility is a useful identifier for pathogens. So if you have an infection and you go to the doctor, they take a sample and they send it to the microbiology department in the lab. If what they're trying to do at that point is identify that pathogen, they want to know what bacteria you have that you're infected with, so they know the best method of treatment. So there's lots of different ways that we can narrow down by process of elimination the identity of a microbe using various what we call chemical reactivity identifiers, but one of them that we can do is motility, because not all bacteria are motile. So if we have what we call a semi-solid motility agar, and we were to take a sample of the bacteria on a needle, this is an inoculation needle, so you should have seen that in lab. But if we take some on a needle and stick it into the tube and pull it right back out, if the organism that we inoculated the agar with is non-motile, then this is what we're going to see. However, if we did the same thing here and the organism is motile, you'll see that the whole agar will become cloudy because it will disperse throughout the tube. So this would be an example of a non-motile organism. This would be an example of a motile organism. And again, since not all species are motile, it narrows down what bacteria you could potentially be infected with. All right, and this shows you an example of Listeria monocytogenes, and I thought this one was of interest because it does a little weird thing with its motility. It has this little umbrella shape. Kind of cool. All right, next, let's talk about some additional appendages. There are the possibility that the bacteria can have fimbriae, pili, and nanotubes or wires. Fimbriae are like bristles. They're like bristles. So this would be an example of fimbriae here on the green that's extending off the bacteria. And you can see them, it's the fuzzy area around, that dark gray. There's a lot of again, it's bristle-like, so you can't really see the individual strands here, but there's lots of little bristle-like hairs, sort of, that you'll see on these bacteria that have fimbriae. The purpose is to aid in attachment, so know the purpose of the fimbriae. It helps with adherence to surfaces, so it helps stick to your cells better, so that they have a increased ability to infect a host, which is what I mean by virulence. How easily is it for it to get you sick or to infiltrate your body, I guess is a better way to say that, because remember, not all bacteria are going to be pathogens. So next we have conjugation pili. These are tubes, they're channels that bacteria can grow in between cells. So here we see pili. This is a tube that extends from one bacteria to the next, and they polymerize these tubes because it allows them to share DNA. Typically, bacteria, when they divide, when they produce more cells, mitosis, y'all should have heard that in A&P or biology. Technically their process is called binary fission, but it's similar to mitosis in a way where it's basically the cell duplicates its genetic material and then divides. So that doesn't lead to any genetic variation though. The genetics of the progeny will be the same as the parents. However, we can see genetic variation in bacteria due to these conjugation pili. They can exchange genetic material between one another, and that's pretty cool for them because it allows them to incorporate new genes, some of which are antibiotic resistance genes. So that is definitely very clinically significant because we have bacteria out there nowadays that we cannot treat with antibiotics because they are resistant to everything. And finally, some bacteria form nanowires or tubes, which are also channels, but it's to share nutrients instead. All right, let's move to our external structures, the S layer. That is this one right here in that, I would say, blue color. It is a layer of proteins that can be produced by some bacteria if they're in a hostile environment. And so that means, if it's a really dry environment, if it's nutrient depleted, etc. But this kind of acts like a bacterial armor. And again, not all bacteria have them, some do. The glycocalyx, that can be in two different forms, a slime layer or a capsule. That's going to be the external green layer. Slime layers are called slime layers because it is sticky. it protects against a loss of water and nutrients. Capsules are denser and they're very thick, and they protect our bacteria from the immune system. So these tend to be ones that cause disease more often. So for example, this is showing you a blood agar plate with a bacteria that's growing on it, and then they also showed you a light microscope image of it looks like a capsule stain to me. So this would demonstrate that white around it, that where the stain didn't penetrate, that is the capsule around these rod-shaped bacteria. Now this is also important because it allows for organisms that have a glycocalyx to form biofilms. So biofilms, if we break that down, living thing and film. When it says film, it's not talking about the movies. We're thinking of, like a film, a sheet of something that's covering it. And so, for example, plaque on your teeth, they're an example of a biofilm. The plaque on teeth protects bacteria from becoming dislodged. These biofilms are responsible for persistent colonization, meaning reinfections, of plastic catheters, IUDs, metal pacemakers, and other implanted devices. So we got to make sure, when we put those in, that they're sterile, because then it can cause this persistent infection that's very difficult to get rid of due to the ability of these bacteria to form biofilms. At the bottom here we see some illustrations of a biofilm forming. So here's the surface of something, some kind of organic surface, a living surface. Let's say it's something in the body, like your teeth. We'll go with that example. We'll pretend that's a tooth. And you see these first little bacteria that begin to stick to the surface. Well, that glycocalyx, again, is sticky, and it helps, it really assists in adhesion to other bacteria. So now we see this sticky mess of bacteria that are sticking together. Then additional microbes are attracted to that, and now you're seeing all kinds of organisms here that are embedded into this glycocalyx sheet here, this biofilm. And that can be very difficult to treat, once again, because now these organisms are protected by this biofilm. And as you can see here, lovely infection due to a biofilm in the body. My arrow looks pretty crappy there. There we go. All right. So next, let's talk about the cell envelope. This includes the wall and the cell membrane. So our cell envelope is composed of 2 or 3 basic layers that perform a function, but they act together as a single protective unit. So when I talk about the cell wall and membranes, I kind of like to tell students, imagine it like a water balloon. The boundary of that water balloon, the rubber, is the cell wall and the cell membrane. So if we list the layers of this envelope from most internal to external, we have the cell membrane, also called the cytoplasmic membrane or the plasma membrane. I know that's a pain in the butt because there's multiple names for the same thing, but I can't help it. Then we have the cell wall, that's a little bit that lies on the outside of the cell membrane. And then we have an outer membrane in some bacteria, specifically gram-negative bacteria, and you will learn what that means later. So let's look at our cytoplasmic structure first. Our cytoplasmic membrane is made up of a phospholipid bilayer, and I really hope that y'all remember this from Biology 1 or A&P. But this cell, what this is demonstrating here, is the phospholipid bilayer, the plasma membrane. And if we wanted to break it down, these phospholipids look like this. Okay, so we have what we'll call a phosphate group, and it then has on the other end fatty acid tails. Interestingly, this side of the molecule is polar, and this side is nonpolar. We won't get into the details of what that means. If you took me for biology, you should know what it means. But what I will say is polar things are hydrophilic, means it likes water. Nonpolar things are hydrophobic, means they don't like water. So these phospholipids, when they aggregate and form a cell, they're going to do so in a specific way. That way is going to be where those fatty acid tails don't touch water and the polar heads do. So if they sandwich themselves together like this, we're seeing multiple rows here of many phospholipids, you will notice that they are going to have their tails point towards each other, and the heads will point towards the extracellular side and the intracellular side. So taking that all together, the big picture here, we have a cell, and it's showing you a cross section here. We see that the fatty acid tails are sandwiched between the polar heads here and here. So the polar heads, the round balls, are facing the intracellular side as well as the extracellular side. And the reason for that is because most of what's inside the cell is water. Most of what's outside and in between cells is water. So those polar heads like that, and those nonpolar tails, they do not. So they get away from water. So anyway, this barrier here is essentially what separates the internal and external environment of these cells. It's sort of like their skin, and it is selectively permeable, and that means it only allows certain things in and out of the cell. Not anything and everything can just go in all willy-nilly. The cell decides that based on, we'll just say based on chemistry for now. Alright, so I have a video here. I want y'all to watch the video. Due to time's sake, I don't want to make this video, I want to make it as short as I can, but I want y'all to watch this video. That way you can get a better idea of how the cell membrane is structured, and we can move on if you're feeling confident. So some additional things that are inside of the plasma membrane are proteins. So the plasma membrane is embedded with lots of proteins. That's these little purpley red guys that are dispersed throughout. They contain cholesterol as well. So I don't see that one pictured in here. It is in the video, I believe, but there are cholesterol molecules that are found within the lipid bilayer. There's glycolipids and other things. The cell wall is made up largely of peptidoglycan in most bacteria cells, and that's very important, hence why I have it all in red and underlined. And peptidoglycan is a repeating framework of sugar chains cross-linked by protein fragments. So I don't expect you to memorize all of this biochemistry, but I do want you to just stick with me here so I can show you what this means. So the glycans are sugars. Sugar chains are the glycans, which are M and G. So you can see they are literally linked together. And then we have a second linear linked together chain of glycans, a third, fourth, and fifth. Then these glycan chains are interlinked with each other by this green line. And that green line is the interbridge. And the interbridge is essentially amino acids. There we go. Alright, so that is what peptidoglycan is. And again, I mentioned this in the previous slide, but note you do not need to know, and I meant to put fine, sorry, these fine details, but you do need to know about peptidoglycan. You need to know that it is in the cell wall. Very important. So now let's move to additional outer membrane layers. These are found in gram-negative bacteria cells, and you will find out what that means later. But again, if we have a gram-negative cell, they're still going to have a plasma membrane. That's in green. They're still going to have a layer of peptidoglycan, but these also have a blue layer we call the outer membrane. So this picture is a very simplified version of what we're seeing in this picture. So this here would be, this pink would be the inside of the cell. We see the plasma membrane here. We see our layer of peptidoglycan, and because it's a gram-negative cell, there is an outer membrane as well. So it resembles the cell membrane because it is a layer of phospholipids, or two layers of phospholipids that are sandwiched together. But it contains some interesting chemicals that we need to know. It contains LPSs, lipopolysaccharides. So these are represented by these black squiggly lines. They're embedded throughout the outer membrane of gram-negative bacteria. And this is important. This is very important because it is going to cause these bacteria cells that are gram-negative to be more difficult to kill. So they're harder to treat in the clinical setting than our gram-positive bacteria. So we've been talking about gram-positive and negative throughout this, and you're probably, I'm sure, wondering, what does that mean? So now we're going to talk about it, now that you know the varieties of cell structure within the wall. So there's two main varieties of cell structure, and they are distinguished by the gram stain, hence the name gram-negative, gram-positive. This was a staining method that was developed by a man named Hans Gram over 100 years ago. And to this day, it's still the most widely used microbiological stain in any clinical setting. So there's two possible outcomes when you perform a gram stain on bacteria. They're either going to turn purple, like some of these guys, or they're going to turn pink, like some of those guys. So here we see a mixed culture, meaning we have two or more different species of bacteria here, and some are gram-positive and some are gram-negative. Alright, let's compare the cell wall of our gram-positive and gram-negative microbes. If we look at the gram-positive cell wall, it has our inner membrane, of course, their plasma membrane, like all cells have, and it has this really thick layer of peptidoglycan in purple. Notice over here the gram-negative cell wall has a very thin layer of peptidoglycan. So this layer of peptidoglycan in our gram-positive bacteria is huge, okay, very, very thick sheet of peptidoglycan. Looking at our gram-negative cell wall, again, the peptidoglycan layer is very small, and of course we also have our outer layer that contains those lipopolysaccharides. Now that's important because the cell wall structure affects how the cell will react during a gram stain. So the steps of a gram stain are as follows. You will create a slide of bacteria. We're going to do this in lab at a later date, but you would create a mount of bacteria on a slide, and you would apply the following items to it in this order. You would first apply crystal violet. It's a stain, and it's a purple color if you couldn't tell by the name. We'll wait 30 seconds. We will rinse it with water. We will then add grams iodine, which is a mordant. So this is a stabilizer that causes the dye to complex with the peptidoglycan. Essentially, it helps it stick to the bacteria. Then after we have given iodine to the smear that we have created on the slide, we will rinse it, and then we'll apply a quick application of alcohol and rinse it, and then we will finally add the safranin, and that is a red dye, to our slide that we have our bacterial sample on. We'll wait 30 seconds, rinse it with water, and view it under the microscope. So if we have a gram-positive bacteria or a gram-negative bacteria, that crystal violet is going to interact with the peptidoglycan and turn the cells purple. The iodine will help the crystal violet stick better to the cells, but remember that our gram-positive organisms have a thick layer of peptidoglycan, so they can hold in a lot more crystal violet and it sticks to them better than it does our gram-negative counterparts. So after we rinse and then wash with alcohol, That alcohol is going to dissolve the lipids in our outer membrane that gram-negative cells have, and it removes that dye of peptidoglycan in gram-negative cells. So after we apply alcohol, our gram-positive cells remain purple, and our gram-negative cells are now colorless. Safranin is then added, and if you add pink or red to purple, it's not really going to change the color. But if you have a colorless bacteria, such as the gram-negative cell at this point, and add that red dye, they're going to stain pink. So that's why you see gram-positive cells purple under the microscope, and gram-negative cells will be pink. Alright, so again, I have a video that goes into the difference between gram-positive and negative that you can watch on your own time, but I will skip it for the sake of time. So let's look at some examples of bacteria that cause infections in these categories. A few examples of gram-positive organisms that can cause infections are Streptococcus, and those would be circular bacteria that form chains, right? Because strepto means chain, coccus means circular. Enterococcus and Staphylococcus. And we know these are all cocci, right? So they're circular. Our staph form grape-like clusters, so they would look like this. Some gram-negative examples would be Klebsiella. It is a gram-negative rod. Acinetobacter, I'm pretty sure that's a gram-negative rod as well. Pseudomonas aeruginosa, that's my favorite word, by the way. Also gram-negative rod, and so is E. coli. I have the hardest time saying the long name for the E there, Escherichia coli. So I usually just shorten and say E. coli. Alright, so now remember I said that bacteria cells are known for having peptidoglycan in their cell walls. You need to know that. Well, there's always exceptions to every rule. Some bacteria do not have typical cell walls, and some of them have unique lipids that make up their cell wall called mycolic acid. Mycolic acid makes these bacteria very resistant to stains, such as the ones that are used in the gram stain. So if you do a gram stain and you don't see any bacteria, even though you know they're there, well, then that might tell you that it's one of these atypical cell wall individuals. So we can try a different type of stain called an acid-fast stain. And it uses a couple of different dyes, a primary dye and a background dye. But the bacteria will stain pink or red. So what you're seeing in this image here, the blue means nothing. It's just the background. But the pink, that is gram-negative rods. Or excuse me, I didn't mean to say gram-negative. I'm still thinking about the gram stain. But these are acid-fast rods, which means that they have unique lipids of mycolic acid in their membranes and walls. So some examples would include Mycobacterium and Nocardia. Another example of a non-typical cell wall is some bacteria don't have a cell wall, which is unique because most bacteria do. But one group of organisms that don't have a cell wall in our domain bacteria are mycoplasmas. They have other protective mechanisms to prevent the cell from lysing or rupturing, called sterols. An example of an organism that is a mycoplasma is Mycoplasma pneumoniae. This causes what we call a walking pneumonia, not to be confused with pneumonia, the generic pneumonia that you might I'm sure y'all probably heard of. But they call it walking pneumonia because these patients that are infected with this bacteria typically don't have the severe symptoms that someone that has traditional, we'll call it, pneumonia does. They still can be relatively active. But what pneumonia is in general, a lot of people say, oh, well, they caught pneumonia. And that's not really a correct way of using that word, because pneumonia itself isn't infectious. It isn't infectious. It's a syndrome. It can be caused by a variety of different bacteria or viruses, but what pneumonia basically is if we look at the structure of the lungs. So you A&P people should recognize this. Biology, maybe not so much, but I'm going to tell you about it. The lungs, if we look at them microscopically, they're made up of millions, trillions maybe even, of these little air sacs that we call alveoli. And they are all intertwined with the cardiovascular system, which is your heart and all the blood vessels. So as you can see here, this one shows it very clearly, that there's blue and red lines going around them. That's representing your cardiovascular system, the capillary bed there. And then if we look at here, it's showing a cross section. So what we're seeing is this little alveolus that's filled with fluid. And we see our cardiovascular system as well, capillary bed where gas exchange will occur. We'll have oxygenated blood that is designed to pump the oxygen, we'll have deoxygenated blood that is here to receive oxygen from the lungs and will deposit carbon dioxide in return that we breathe out. So let's look over here now. Here we have a nice normal alveoli. We see flow of oxygen and carbon dioxide that's happening all nice and pretty like it should. But if we look at an unhealthy alveolus, we see this fluid. That fluid prevents this movement of gases in the lungs. And of course that's not good because we need oxygen to survive. Now moving to 3.4, we're going to internal structures. We have the cytoplasm. That is the fluid in the cell. It's mostly water. We have the nucleoid. That's the region here where DNA is found. Not to be confused with nucleus. Some bacteria often have plasmids. These are accessory rings of DNA, and I say they're accessory rings because they're non-essential. They don't contain struggling here. They don't contain genes that are essential for life for the bacteria. So it's extra stuff like resistance to drugs or toxins, being able to make special enzymes they don't need to survive. Next, bacteria have ribosomes, one of the very few organelles that they do have. And you should know that these are sites of protein synthesis. They consist of a large and a small subunit to formulate the entire ribosome. And what they do is they read mRNA and then they make a protein out of it, which is something that my biology people better remember, and my A&P people I don't know if y'all took it with me or not, but y'all should be pretty well comfortable with that as well, I hope. If not, review. So these ribosome subunits are characterized by what they call S units, and if I recall what that stands for, it's and I'm going to butcher this, but Svedberg units. I don't really need y'all to memorize that. I just know that I might have some people ask me about it, like why is it that 50S is the measurement for our large subunit and 30S is the small subunit, but together it's 70 instead of 80. So this isn't a simple addition of small and large subunits to get that S. Because the S, the Svedberg units, it's in reference to densities, and we're not going to go into the details, but basically what I do want you to know is that bacterial ribosomes and eukaryotic ribosomes do have different measurements in their Svedberg units. So bacteria is 70S, eukaryotes are 80S, and why that's relevant in the clinical world is because drugs can be formulated to target 70S ribosomes and therefore target bacteria. Next, we have inclusion bodies. So these can be used for food or gas storage. They can store other things too. There's some interesting articles about those and types of bacteria called metotactic bacteria. I'm not going to test you on this, but if you're interested like I am, you can watch this video here. We also have bacterial microcompartments. These were recently discovered in the 2000s, like that's basically yesterday, I feel like, in the microbiology world. But these little compartments are packed with special enzymes that work together to do some kind of biochemical pathway. So these are almost like functional analogs of the lipid-bound organelles that we see in eukaryotes. Pretty neat. Next, we have the cytoskeleton. If you break that down, we have cyto means cell and skeleton means, well, skeleton. It's essentially the bones of the cell, if you will, because it gives it its shape, just like our bones give us our shape. They have also been identified in archaea, but they're proteins. The skeleton, quote unquote, is made up of proteins that will extend throughout the cell that will help to give it its structure. So what you're seeing here is a confocal microscope image using fluorescence technology of the various types of tubules. There's filaments and tubules, we'll say, that you'll see in the cytoskeleton. There we go. Next, some bacteria can have endospores. Not all of them, but some of them. And what those endospores are is they are a protective variety of a bacteria when they're exposed to harsh conditions. So under normal, happy conditions, we have bacteria that we call vegetative cells. They're doing their thing, living their best life. If they come into contact with a hostile condition, then they will sporulate, meaning they will form a spore. And that spore will be dormant. It will stay dormant and protect the cell until favorable conditions come back again. And so, interestingly, this can protect the bacteria for many, many years. So endospores have been recovered and revitalized. We put these bacteria in nice conditions, and they were able to be revitalized as old as 250 million years. That is insane, but really cool. These endospores resist extreme heats, drying, freezing, radiation, and any other chemicals that would normally kill the vegetative cell. So it's almost like this bacteria just throws on a coat of armor and hangs out until it's safe. So this shows you a step-by-step cycle of sporulation and germination. So here we see a nice vegetative cell. It's becoming depleted of nutrients, and it's like, oop, okay, I really need to sporulate because I'm not in a good condition. And so you'll see over time, we'll have our early endospore forming, and eventually the endospore is what we have here, and everything else is in the core, and it stays in that little safe little bubble almost. It's almost like it kind of reminds me of a seed, even though it's not a seed, but it's going to stay there until it reaches a spot where it senses that it's got a nice place to be in. And once it returns to some kind of nice place and the weather changes or whatever the case is, it will germinate. So we have vegetative cell, sporulates, and then the spore will germinate. Oof, it's really hard for me to write with this thing. Or germinate whenever it's in happy conditions. Alright, so let's look at some bacteria of medical importance. So one that has a endospore, that forms an endospore, is Bacillus anthracus. This is the agent that causes anthrax. It is an agent of bioterrorism. Some of y'all may remember back in 2001 there were anthrax attacks in the United States through the mail. And some individuals that are in politics, like some Democratic senators, were sent letters that contain anthrax. And this, basically was in a powder, and it aerosolizes. So, it was... I guess you could say it wasn't able to be prevented because they didn't know. But due to that, it killed 5 people and infected 17 others. Without treatment, inhalation of anthrax is almost always fatal, but with aggressive treatment, about 55 patients survive. That's still not good odds. So what you're seeing here are anthrax bacteria, the little gram-positive purple rods. Another example, Clostridium tetani. This is the cause of tetanus, or you may have heard it as lockjaw. What this bacterium does is it has the capability of producing a neurotoxin, and it causes these uncontrollable, painful muscle contractions, particularly in the neck, and this can be deadly. So this is a picture, a painting from 1809, where you can see this individual has some very odd shape to his body. That is because he is experiencing tetanus, and here we have an actual picture of a person experiencing the same thing. Even with treatment, about 10% of people who contract tetanus die. Luckily, though, it is vaccine preventable, so get your Tdap. All right, how do these look under the microscope? Pretty neat, because they look different. They're gram-positive rods, but because they are capable of forming spores, sometimes you'll see them look like tennis rackets under the microscope. Another example is Clostridium perfringens. This is one of the most common causes of foodborne illness, and I put this in here. I thought it was funny. There is a outbreak of Clostridium perfringens at Chipotle. I don't remember how long ago it was, but you see all the ambulance coming through. Is it E. coli or salmonella? Well, it was Clostridium perfringens. So I did write in when it was. I was in 2018 in Ohio. They have they are a rod shape as well, and they have a terminal spore. So it almost kind of looks like our tetanus, but not quite. Now it's a nasty bug. Clostridium perfringens is the third most common cause of food poisoning in the United States, and it's also the causative agent of gas gangrene that can appear after surgery wounds. So that's what you're seeing here and here. And I know that's pretty not nice to look at. But I also wanted to make this a little happier, so I put in this image. And if you're, even close to my age, maybe, you might remember watching the Powerpuff Girls whenever you were younger. And I always thought what they were saying was the gangrene game. And in reality, let me relate this to this picture here. So growing up is realizing that the green villain gang from the Powerpuff Girls was called the Gangrene Gang and not the Gangrene Gang due to their gangrenous green skin. And honestly, I feel like my third eye just opened, because when I was a kid, I did misspeak whenever I was referring to them. I called them the Gangrene Gang instead of the Gangrene Game. Who knew? All right, next, Clostridium botulinum. This bacteria is the cause of botulism. And what it does, what it has the capability of doing is making a neurotoxin that can lead to paralysis. And if you have paralysis to your respiratory muscles, you can't breathe, and you can die. That's about 10% of the time. So pretty high rates of mortality there. This bacteria is associated with dented cans. So I don't know if y'all's grandma ever taught you never to buy dented cans from the grocery store. Mine did, but hey, maybe yours might have told you to get the dented cans. Maybe you can get a discount. That was my mom. Anyway, so also they are associated with improper canning techniques. So these are gram-positive spore-forming rods that also look like tennis rackets. Another one that's really important, Clostridioides difficile. Usually in the hospital setting, they just call it C. diff. Very, very, very important in the clinical setting because it is the most common cause of nosocomial infectious diarrhea. Nosocomial means hospital acquired. Acquired. I think I missed a C in there. Close enough. Hospital acquired. It means you got the infection from the hospital. So yeah, sure, hospitals are, no, we got all the medications here. We're going to make you feel better. But who goes to a hospital? Sick people. So naturally, it is very possible when you walk into a hospital that you can get sick from just going there because it's a hub for sick people. So the frequency of colonization in hospitalized patients varies from 10 to 43%, and that's a big deal. Hospitals are very concerned about that. Um, why does this happen? It typically happens because our normal gut flora, we have normal bacteria in our gut that is eradicated when we treat with antibiotics. So if we treat with antibiotics and we wipe out our gut flora, then it's more likely that we can have some really nasty bacteria that can persist and then cause infections such as C. diff. So 1 in 11 people over age 65 are diagnosed with healthcare associated C. diff infections, and they die within 1 month. Now, pretty intense. Very, very important. I bet if y'all have worked in clinical settings, you've probably heard of it before. So that is it for our chapter 3. So I'm glad I made that only an hour long. So I hope y'all enjoy it and make use of it, and I will talk to you guys later.